Zoom recording. Okay, there we go. So a bit about us again, Women in Aerospace Medicine. We are associated with AMSRO, which is the student and resident organization of asthma. And we have a vision, which is to advocate for women and women identifying persons in aviation space and environmental medicine. Among students, residents, and also we have some doctors and scientists that are a part of WAM as well. We welcome people of all identities, and we just strongly advocate for women and women identifying people, and we started out as the diversity initiative of AMSRO, and we continue to advocate for diverse groups in aerospace medicine. So we launched in 2020 and now we're in 2024. So that's 4 years. Very, very exciting. And here is some of our leadership. And you can see, I don't know if you can see my mouse, but I'm on the left and I'm Molly. And this is Sarah and the rest of our leadership group. A few updates. So reminder that asthma, the big conference, is happening in May. And it's in Chicago, so go to the asthma website to learn more information about that. I'm going to admit some more people. Also, we have some bylaws that we have been working on. Specifically, Sarah has been working very hard on these bylaws for WAM. And we also are going to have some upcoming leadership opportunities, and we will send out the application form in the next month for different positions in WAM. And also we are planning a WAM in-person event during the asthma conference. So stay tuned for more information on this event. And again, I would like to introduce Dr. Sigrid Reinsch. And please put any of your questions in the chat. You can leave your camera on or off, but note that we are recording the meeting. And mute your microphones while the talk is going on. So thank you so much, and would love to have the speaker begin. Thank you.
Great, I'll share my screen. Does that look good?
Yep, looks good. Thank you.
Great. Okay. Yes, I am very happy to be here today. Thank you for the invitation. My name is Sigrid Reinsch, and currently at NASA. I've been there for more than 25 years, but currently I have two hats that I wear. One is the project scientist for NBISC, and then I'm also the director of the SHINE program, and I'll tell you about both of those things. So just feel free, I will try to make this brief, and then we can have a much longer discussion. But I'll tell you a bit about, let's see if I can get these to go. Okay, so I'll give you a bit about my background and education and my path to NASA, and then I'll talk about the SHINE course, which is a space radiation virtual course. Also, we have a practicum that we're running this year, and then about NBISC, which is NASA's Biological Institutional Scientific Collection, and then any questions that we have. So just to start off, I just really want to say that coming to NASA was not anywhere in my goals. When I was a kid, I'd never dreamed of being an astronaut or anything like that. In fact, when I got the job, I really felt like, a round peg in a very square hole. And this is basically how I got my job at NASA, was due to the fact that I had worked on frogs for many years, and in particular frog eggs and embryogenesis. And they had just recently done this frog experiment. And so I was able to, really understand the significance of that experiment. They also were looking for people with my expertise, which was in the cytoskeleton and cytoskeletal proteins. So I just happened to be, kind of at the right place at the right time. I was really looking for a job, so that also, really, I had looked in academia and hadn't been successful, and so this was a nice compromise, which I'll tell a little bit more about. So I was applying from Germany, where I had done my postdoctoral fellowship. So here's my background: is I did my training at UC Santa Cruz and got a BA in biology and plant biology. And then I went and did a stint at Brown University for several years and worked on chicken retroviruses. I then went to grad school at UCSF and worked in cell biology and worked on Xenopus microtubule cytoskeleton and neuronal development. I went to EMBL in Heidelberg, Germany, continued my cell biology, working on Xenopus motor proteins and nuclear positioning. In particular, how does the two pronuclei in a fertilized egg get together, which is in a big egg like that's 1 millimeter in diameter. That's a big question. And then since 1998, I've been at NASA Ames. And so, as I said, my expertise is in cell and developmental biology and molecular techniques, microscopy, non-mammalian vertebrates, including xenopus and zebrafish, and invertebrates, tardigrades, and then the cytoskeleton. So currently, as I said, I'm the NBIS project scientist and the director of the SHINE radiation course. Previously at NASA, I've had a lot of different roles. I started with the Xenopus tropicalis insertional mutagenesis project, which never really got off it got off the ground, but I ended up losing the funding for that. I've worked on tardigrade development and resistance to radiation. I worked on the NASA Omega project, which was a biofuels and algae project. I've been a Gene Lab senior scientist for many years. I continue my affiliation with Gene Lab. And then I was the director of education and outreach for the space biology program. And for 3 or 4 years, and during that time, I was the director of the Space Life Sciences Training Program, which is for undergraduates, and the director of the Gene Lab for High School program, which is a great omics program, teaches omics to high school students. So let me delve into my current role. So I'm the director of the Space Health Impacts for NASA Experience, or SHINE, space radiation curriculum. That program will start on this coming Tuesday in its second year. And basically, cosmic radiation, as many of you know, is quite a huge risk for astronauts that are traveling in deep space. So on the ISS, it's not that big of a risk because the ISS lies within the Van Allen belts, which you see. I don't know. Can you see my mouse? Yes, no?
Yes, we can.
Okay, so these are the Van Allen belts. They're caused by the Earth's magnetic field, and they basically trap the radiation from the sun, which is largely protons, and then they also will trap the other cosmic radiation, which is largely more heavy ions that are really do a lot of damage. And so there are a few of those that do get in on the ISS, but once we go to the moon or Mars, we're going to go beyond those, and so we won't be protected. And so, so the background for the course is that there had been a space radiation summer school, which was in person for 3.5 weeks at NSRL at the NASA Space Radiation Laboratory in Brookhaven. And that ran from 2004 to 2017. It was really a wonderfully successful course. And it covered radiation physics to radiation biology, and the students really got a hands-on experience in doing experiments in the beamline. Another predecessor to the course is the Spaceflight Technology Applications and Research, or STAR program. This is a course which is just completing this week. They're completing their 4th year. It's a virtual NASA program that was originally designed to be in person at NASA Ames, to basically train principal investigators and senior research scientists and postdocs in how to do a spaceflight experiment. And that happened, the first year happened right at the beginning of the pandemic. And so it was quickly converted into a virtual course. And that virtual course allows international participants. It's an absolutely fantastic course. If you haven't heard about it, I urge you to apply. The applications will open pretty soon, and it runs every September to February. It originally started with 20 participants, and now it takes about, sorry, just trying to see if I'm getting messages. Sorry. It takes, now I think they're up to taking 25 or 30 participants. So they've expanded the number of participants that they're taking. So back to the Shine program, it's basically a virtual program as well. It'll run, this year it's running from February through the end of July. Runs weekly, about 2-hour sessions, and it aims to educate the participants in the physics, the scientific aspects of space radiation, as well as the agency's risk management strategies. We also do a lot of networking, and there is a requirement in this course, which is different from the STAR course, which doesn't have any requirements except attendance. But in the space radiation course, what we have the participants do is to actually develop a beam time proposal to actually do an experiment at the NSRL. And one of the things that we do is we can have the participants actually pair with each other or form groups based on the topics. And that actually really has been a wonderful way for them to network with each other, to share their knowledge, to share their expertise in different areas. And then in this fall, we're going to have an actual practicum session at NSRL, and 5 participants will get to go to the NSRL and get some hands-on experience. So the Shine Space Radiation Virtual Program, we have a solicitation through NSPIRES. If you don't know what NSPIRES is, ask me at the end, and I'll put a link in the chat. And that solicitation is open for a month or 2, and then we sent the invites in December. We actually, some of our course members attended the Human Research Program Investigators Workshop, the IWS, in Galveston. And so they were able to meet each other as well as some of the speakers. And we also had alumni from last year's program that attended that meet and greet, which was great. So this year we are going to have 26 participants. They're from, let's see, 16 of them are actually foreigners, and about half are from the US. There's a number from Canada and Poland and France and Korea, South Korea, and several from African countries, from Cameroon and Nigeria, and several from India. So it's just, really a great international group. Many of those who are foreign nationals are actually based in the US currently, or based in other countries. So these are some of our speakers. I'm not going to go through all of their names, but we have this huge number of speakers that present each week. So each week is on a different topic. And then the participants, these were the demographics. So this course is open to graduate students, and then research scientists, anybody from a graduate student on up. And last year we had expertise in all different topics: molecular and cell biology, botany, microbiology, rodent work, biostatistics, planetary science, and aerospace medicine. We had modelers and epidemiologists. So it's really, really a great mix of people and expertise and each, participant brings their unique knowledge to the course, which really kind of makes it a much more interesting course. So these are the participants. I'm not going to go, I'll just, flash them up there. These were from last year. I don't have the photo in there from this year, but it looks equally diverse, a very good mix of men and women and different ethnicities. And then, as I said, this is a picture of the NSRL, the NASA Space Radiation Laboratory beamline, where you can do space radiation experiments that are simulate the galactic cosmic radiation. And so a big focus of the course is actually understanding how you can mimic that space radiation to be able to run experiments that can model that and give us results on the physiological or the physics effects of space radiation. And then what they had to do with this proposal was they make a group, or they could do it individually. They had to do an oral presentation of their proposal, and then they had to do a written proposal. And that written proposal is reviewed by the space radiation element scientists, who provide feedback on how well they did on that proposal. So it's not a grade. It's basically, this is a real life activity. These written proposals are the same that if you actually get a grant that involves using the space radiation facility, you have to actually submit a proposal. So it's a real life experience writing the proposal and trying to understand how to calculate how much beam time you need for a particular exposure. So as I said, in 2024, we have, again, the didactic radiation curriculum, which is the virtual component. And then we have the space radiation practicum, which will be held in October. And both of those, we've already selected the participants. We'll put out another call for both of these programs in probably August of 2023 through the NSPIRES mechanism. And then this is just a picture of the linear accelerator. This is the actual lab part. So the whole linear accelerator is this whole part, and then a part of the beam shoots off to the NSRL, which is focused on the experiments. And basically you put your experiment into this beam line for exposure. This is the SHINE team. So I'm the director, and then these are the space radiation element scientists. Robin Elgart has left. So Janice Jay-Z is actually helping me lead the course. Greg Nelson is a huge part. Brock has also left this year. And so we have another scientist, Sergio Santa Maria, who just ran the BioSentinel satellite experiment. And so he's joining us to help lead the course this year. So my other hat is as the project scientist for NASA's Biological Institutional Scientific Collection, or NBISC. And so this is part of NASA's Biological Open Science Resources. And I don't know if many of you are familiar with that, but basically it's the way that NASA, and in particular space biology and the Human Research Program, have trying to maximize the amount of information and scientific discovery you can get from every experiment. And so what happens is, if there's an experiment, for instance, one of the rodent research experiments, there's a PI for that experiment, and that PI has proposed to do a certain set of experiments with those animals. They're looking at something in particular. They may have mutants, they may have a drug treatment, etc. And they get the first dibs on all the tissues from those experiments, but the rest of the tissues actually belong to NASA. And so NASA has a biospecimen sharing program. These are scientists that are very well trained in dissection and preservation of tissues, and they go in and they help coordinate all the dissections from these large experiments. They collect all of the tissues. This is for both flight and also for ground investigations. And they coordinate the internal tissue sharing, and then any of the tissues that are harvested that don't go to the original PI team come to us, to the NBISC project. And so we actually house all of these tissues and then make them available to the scientific public for further research. So it's a collection of non-human specimens, and also we have microbial species that are from space experiments and other types of experiments that are space relevant. The other parts of the open science resource are the Ames Life Sciences Data Archive, or ALSDA. And that goes hand in hand with all of the experiments. They provide basically all of the experimental data, the mission project and imaging data, any assays that are run. They have a whole series of assays that they have standardized, and they're basically moving towards being able to mine the data with machine learning and AI/ML. The other part of it is NASA Gene Lab, and I was part of Gene Lab for many, many years. I still actually function with their analysis working groups, and so they have a wet lab that actually takes some of the tissues that are collected from the scientific experiments and process those and make that omics data available to the public directly, even before publication. And so that's a huge way to generate data that anybody can analyze. And we've had hundreds of papers that have come out of that type of analysis from the data that is stored through Gene Lab, the omics data. So back to NBISC, as I said, it's from space-based experiments and then also from ground analog experiments. Like these can be things that mimic space radiation or the microgravity environment or hypergravity. So those are some of the types of ground experiments that are there. As I said, this really fills spaceflight knowledge gaps by increasing the scientific return on every mouse that's flown to space, or any public money that has gone into running these experiments. And one of the things that NBISC really helps is that because we can provide these tissues to early scientists who don't necessarily have the expertise to run a spaceflight experiment or haven't gotten that experience, they can actually capitalize on these tissues and analyze spaceflight tissues, and it really helps. So we try to make sure that any early career PIs, know about this great resource that they can use to obtain tissues for their analysis. So currently, we have a lot more than we probably have about 40,000 non-human biospecimens that are from space biology experiments, and those are mostly mouse, rat, quail, and microbial isolates. And then we've just taken in a lot of space radiation mouse space specimens, and these are primarily from experiments that were done at NSRL. And so we're trying to get all the data into our database so that we can make those available in the coming weeks or months. And so as you see, we end up getting publications from these tissues. So there's been 39 publications since 2011. I think that number has just gone up. And we get numerous tissue requests each year. There have been the samples that come to us also go to Gene Lab, and so that has generated 200 Gene Lab data sets, which is really fantastic. That's all data that's publicly available for anybody to analyze. The specimens, as I said, are mostly from shuttle and ISS missions. We also have stored specimens from ground analog studies, including centrifuge, hind limb unloading studies, and partial weight bearing studies, as well as those from the space radiation. How do you get them? So the award process is basically for educators and biosciences researchers. We have the NASA Life Sciences portal. That's the current way. That's going to switch over to OSDR, which is the Open Science Data Repository, sometime probably in the late summer, early fall. So the requirements are the requester must be fully funded. So you have to be able to show that you have the funds to be able to analyze the tissues, because we do not provide funding. However, Space Biology did do one call where they did provide funding. And if you respond to a space biology call for experiments and you include a justification for using those tissues as part of your experiments, you can also obtain funding that way. So that's a slightly different process, but we do have a process for directly interacting with investigators and awarding those tissues. So you have to submit a proposal that justifies the request. I'm the first line of receipt of those proposals. I read them through. I see if they're valid. If there's issues with them, I send them back and give you, a critique, basically on how to improve the proposal. So we don't just flat out reject it. We try to work with the investigators to really get these tissues awarded, because I don't want full freezers. I would like all of these tissues out to researchers so that we can maximize the amount of data that we get from these experiments. And then basically what happens if the proposals are approved, they go through a scientific review board. Then the ALSDA actually prepares what's called a research data submission agreement. So you make a formal agreement to actually what kind of data you're going to be generating and how that's going to come back into our open science repository. And once that is signed and returned, then we coordinate the shipment to the PIs. And that can take anywhere from 6 weeks to 2 years, the whole process, depending on how clear and simple the proposal is and whether there is clearly funding for it. So I'm not going to go into all of this, but basically this is how we maximize the return. That we have the primary investigation, the tissues get dissected, the tissues get stored in NBISC, they get entered into what's our laboratory information management system. If we get a tissue request, that gets sent out to the investigators, that data gets analyzed and published, then it comes back, that data comes back to the ALSDA or Gene Lab or our open science data repository, and that data is reused. And so we've seen actually data that from samples that we sent out from NBISC that were actually leftover samples, that then where that data ended up getting put back into Gene Lab and then reused for a tertiary publication. So that's that's really capitalizing on data reuse. So this is our NBISC team. Sam Gibre is the project manager. I'm the project scientist. Joey Varelis does all the nuts and bolts and fulfills all the tissue requests. And Mo Kaze is a PhD scientist who runs our space microbe collection. These are some of our interns that I've hosted this year. They've been helping us with all of our data entry and data visualization tasks. They're really fantastic. And then big thanks to my 2 daughters that have helped me throughout my whole career and supported me. And then all of my siblings. This is a picture of my family. I'm number 10 here out of 14. Let's see. I think these are backup slides, so I'm just going to leave those there and take questions. And let me get back to, let's see, how do I find the Zoom?
Thank you so much for your talk.
Okay, I'm trying to find you. It's somehow maybe you can stop me sharing.
Oh, okay. Let's see.
Okay, I found it. I can stop it.
Thank you so much for your talk. Very, very, very cool stuff. I didn't know that they had a program like that where people could apply and obtain some of the specimens. That's really, really neat.
Yeah, it's really great. We get, as I said, a number of different requests each year. It is a bit of work. We have a template that we send out for the proposal process. So it's not like we provide a lot of guidance, actually.
Okay, Shayna has a question. Can you discuss some likely funding resources?
Yeah, so a lot of early career people, if they have faculty positions where they have startup funds, they can